Yeah. Good morning, all. So, Dr. Shana, shall we start? It's
Yes, sir.
yeah. So, good morning, all, for today's fourth lecture series of modern mycological lecture series in online mode, definitely by conducted by CBEC and MycoAsia together. So, today's topic is applications of MALDI-TOF MS in mycology. A very interesting topic and very applicable in the present times, especially in the medical field. So today's our speaker is Dr. S. Shishupala. Thank you, sir, for joining and taking time out for today's talk. So
Thank
today,
you.
the MycoAsia is, as those who have joined for the first time, MycoAsia is a journal, online journal, which is publishing papers related to taxonomy and applied both aspects from Indian and Asian and other parts of the world. And it has been doing excellent work to promote mycology also through online lecture series and offline lecture series, and trying to educate different parts of the different and trying to cover the different aspects of mycology. CBEC is an NGO working on the diversity and applied aspects of mycology. So we request you to please mute yourself during the talk. Questions will be taken at the end of the talk. Either you can raise your hands or you can write your questions in the chat box. So today's speaker, Dr. Shishupala, is currently holds the esteemed position of Professor and Director of Research and Development in the Department of Microbiology at Davangere University, Karnataka. An accomplished academic, he has an impressive educational background with MSc, MPhil, and PhD qualifications, showcasing his deep commitment to microbiology. Dr. Shishupala's career is marked by his dedication to teaching and research in microbiology, where he has made substantial contributions. And as a leader, he is recognized for his innovative research methods and has played a pivotal role in various academic projects. His work not only enriches the academic environment at the university, but also significantly advances the broader field of microbiology. Professor Shishupala's influence extends beyond academia, and he is deeply committed to mentoring students and young researchers, fostering a new generation of scientific minds. His passion for his field is evident in every aspect of his work and making him a respected and influential figure in microbiology. Thank you, Dr. Shishupala, for joining us and agreeing for this wonderful talk on the topic of MALDI-TOF. With this, I invite you to please go ahead with your talk and share your screen for the presentation. Thank you, sir.
Thank you very much, sir. I take this opportunity to thank MycoAsia for providing me an opportunity, and Dr. Rohit Sharma and Dr. Belle Shanai for this gesture. Thank you very much for an introduction, and I will look for the presentation. Yeah, good morning, all of you. It is seen, right?
Yes, now it is in full mode. If you can hide that notification there at hide, you can click on hide. Yeah, that's all. Please go ahead. Thank you, sir.
Okay, good morning, all of you, and welcome to this presentation. I did work in Indian Institute of Science with Professor P. Balaram for MALDI-TOF through Indian Academy of Sciences. So I have made this presentation in three different parts. The first part will be giving information on principle of MALDI-TOF because many of the students are here. The second part would be the methodology, And the third part would be a detailed analysis of applications in mycology. And it is a great opportunity to be part of MycoAsia group and helping mycology to be developed in India. So this is our university campus. It was part of Mysore University earlier in 1979 and became part of Kuvempu University. And now it is a Davangere University since a decade, 2014. We have 73 acres of campus and I work here in microbiology department. So the topic which I have chosen for today's discussion based on my previous experience is applications of MALDI-TOF MS in mycology. People generally look at MALDI-TOF MS, an acronym, a longer acronym, what it is, how it is used, how it can be used. All that I will be discussing today in another 45 to 50 minutes. So in every aspect of microbiology in general, or biology in general, microbiology or mycology specifically, we need to characterize the biomolecules. It could be proteins, peptides, enzymes, or DNA, RNA analysis, all that is included in biomolecular characterization, which is essential for basic microbiology, microbial identification, and biochemistry like proteomics or genomics, diagnosis of microbial pathogens, whether it is medical microbiology or in veterinary microbiology, then environmental microbiology, and of course, biotechnology and industrial microbiology are offshoots of identifying the biomolecules which are useful for mankind. Then a major concern for biologists is to develop a sensitive method, how minimum sample should be. Sensitive refers to the minimum sample to be required for analysis and to get a reproducible methods of identification of and differentiation of biomolecules. Having said that, a various biological activity guided assays, for example, antibiotics produced, you need to isolate the fungus in a pure culture, then grow and then check the culture filtrate or extracts for antibiotic activity. Like that, enzyme, you have to isolate the fungus, then look for the enzyme by plate assay and then go for enzyme activity. Like that, various biological activity guided assays and screening procedures have been developed earlier. But among them, one of the physical chemical method, but adopted by biologists extensively, is the mass spectrometry. Mass spectrometry will give you mass or molecular weight of a compound or a protein or any other. So it was used earlier by chemists, but later adopted very well by biologists and particularly mycologists, microbiologists, has been used as a potential viable physical method for characterization of biological samples. Then I would like to give a brief background of what is the basic scheme of mass spectrometry, though we may not go into the details for a biologist to understand what are the things available. We have an ion source which is present as a sample chamber where we will be putting our sample and making it ionized. So please remember, in case of mass spectrophotometry, only ionizable molecules can be detected. So ion source, which is the sample chamber where we can place the sample and make use different conditions for making it ionized. The sample molecules getting ionized move in a time of flight tube, which where ions get separated based on their molecular weight and charge, which is referred as a mass analyzer or time of flight tube. Then the individual molecules getting separated from the sample and getting separated based on their MZ, molecular weight and charge ratio, they will be going to the mass detector, which is an output device, which is nothing but a computer which will be putting the result in the form of a spectrum or graphical representation of the molecules found in the sample. So this is an outline of scheme of a mass spectrophotometer. The one which I am going to deal with is the MALDI, one of the methods where an ideal method generally requires a minimum sample preparation. Otherwise, you have most of the morphological or biochemical tests, you have elaborate procedures for sample preparation, microscopy, staining and things like that. An ideal method would, for a characterization, requires minimum sample preparation, permit automatic analysis, as Dr. Rohit Sharma specifically put it. In medical microbiology, you need series of biological samples. It could be body fluids like urine or blood culture or extracts or other things. An automatic analysis would be an ideal one. So it should provide adequate information about the stable characteristics of the organism. Then only it is possible for identifying or the organism or characterizing the biomolecule present. So one such method is MALDI-TOF MS. What does this MALDI-TOF MS stands for? Matrix assisted. That means there is a chemical called matrix which assists the ionization process. Laser being used. So L stands for laser. Desorption. The ions get from the sample get dissolved and they move in the time of flight tube where they get separated, and it is a detection of molecular weight or mass, and hence it is mass spectrophotometry. Matrix assisted. Matrix helps in ionization process. Laser being used. Desorption ionization is the process taking place, and separation of the molecules in the time of flight tube, and then mass being detected, mass spectrophotometry is one of such techniques available for sample analysis. The major goal of MALDI-TOF MS is to vaporize the molecules present in the organism or the sample which you are processing, and ionize the analytes without fragmenting them, without degradation. Most of the other mass spectrophotometers will make use of excessive heat to fragment the molecules. Here, this is called soft ionization, laser being used without spoiling or without fragmenting the sample molecules. Then, by incorporating the analyte with an excess of matrix, as I mentioned earlier, matrix are the chemicals which help in ionization process of the sample. So, the matrix absorb energy from laser and get it transferred to the sample molecules. The sample molecules gets vaporized and ionized. So that is the overall process. That means you have a matrix, a chemical which absorbs the laser energy, provides it to the analyte samples, and make the analyte sample vaporized and ionized. So what are the samples which can be used? It is an amazing to see the literature, and in fact I myself has used it in 2001 when I got a article of using single cell MALDI. One will be able to analyze a single cell, which is generally not available in many of the biochemical techniques, analyzing a microbial cell. So intact cells can be used, tissue samples can be used, virus particles, cell lysates, microbial extracts, body fluids, purified proteins, or can be analyzed within 5 minutes. Oh my God, within 5 minutes. That's why I did mention it is a rapid method. Then diverse samples like air. For example, you want to look at the allergens present in an indoor environment or in a food industry, whether you want to look for contaminants or soil analysis, physiological fluids, clinical specimens, all can be processed and analyzed. Aerosols containing bacterial and fungal cells or spores can be directly trapped with a air sampling device and then used in mass spectrometry directly. So having said that, the sample to be used either in the form of a solution, or for example, purified proteins and others, or the fungal spores are taken in a solution, for example, spore suspension. What is the amount of sample required? It is only 2 microliters, only 2 microliters. So small sample. I was referring earlier, ideal method would allow simple minimal sample preparation and rapid method. So sample solution here, fungal spores, the mycelium is taken in a suspension water or deionized water or other solvents, which I will refer later, then placed on a sample slide. Sample slide here is not a microscopic slide. It is a stainless steel style where you will be able to place a drop and allow it to air dry. This is called dried droplet method. Then one can use mixture of the sample along with the matrix. I did mention matrix are the chemicals required for ionizing the sample, assisting the sample ionization. So that also can be, matrix can be mixed with the sample and directly placed, which is referred as seed layer method or 2-layer method. For example, you have to put 1 microliter of matrix, 1 microliter of the sample, and allow it to dry, 1 more microliter of matrix, 2-layer method or seed layer method. Matrix solutions which are used commonly are alpha cyano 4-hydroxycinamic acid. These are the cinnaphinic acid, genesic acid, ferulic acid. These are the compounds which readily absorb laser energy, transfer it to the sample molecules. That's why these are preferred. Particularly, alpha cyano 4-hydroxycinamic acid is preferred for proteins and peptides analysis. So this is actually the sample plate in the MALDI-TOF. So you can make out there a stainless steel plate having circles. In each circle, it will hold 2 microliter of the sample. One can load up to 400 samples at a time in this plate. So only 2 microliter of the sample is required. Up to 400 samples can be processed at a time. Then this is the instrument available in Indian Institute of Science. This is an horizontal model. You have this part, the left-hand side, the opening of plate, the ion chamber.
Dr. Shishupala, your voice is breaking, sir. Your voice is breaking.
Yes.
If you can switch off your video and then speak.
1 minute, sir.
So, presentation is clearly visible, sir. Only your voice is breaking.
Yeah, I understand. Okay, sir.
Yes,
now,
yes, no, yes, sir. Now
yeah,
it's very clear. Yeah, thank you.
Yeah, the soil, the sample holder is in your left-hand side, and the time of flight tube is conceived inside, and there is a provision for laser beam to come and hit. I will show you in the next slide how the laser beam comes and hits. Then you have a detector which will be output into the computer monitor, where you will be able to see the spectra. So how the basic process? It is an interesting slide here, which I have taken without any audiovisual expertise. Myself, I don't know, but I used PowerPoint to make this slide for a biologist understanding of MALDI-TOF MS. A sample plate, a stainless steel plate, where you are adding sample and matrix solution. Matrix, as I mentioned, the chemicals mixed with the analyte, which will be considered as an analyte. You have a sample there, the intact cell or a body fluid mixed with the matrix. When the laser beam comes and hits the sample, the matrix readily absorbs the laser energy, gets ionized, and making that energy transfer to the samples also, and they get ionized. So when I say ionized, it could be cation or anion, positive or negative molecules. Some are neutral, which cannot be ionized, so they will be staying there only. So laser has hit the sample, the ionization, desorption will occur now. They will leave the sample chamber into the time of flight tube. So what will happen in the time of flight tube? You have certain molecules from the sample are positively charged, certain are negatively charged, certain are neutral. So time of flight tube, you can have an electromagnetic field in order to allow only positive ion mode. So when you allow positive ion mode, only positive ions from the sample chamber will be able to move across the time of flight tube and goes to the detector. How they move? You just see this. So matrix, small molecules, they move further, allowing the positively ion molecules. There the ion separation takes place. What is the basis for ion separation? is the molecular weight. So the smallest molecule will move faster than the little larger, and largest molecule will move slower. So the whole thing occurs within seconds. I'll just repeat this for the benefit. So you have matrix moving, positively charged ions moving, the sample molecules getting separated into the detector. So what happens here? When you have a sample chamber, time of flight tube and detector, the smallest molecule, positive ion molecule, will go to the detector, followed by little more bigger, little bigger in terms of mass. Mass charge ratio it is considered, but here all positively charged molecules, then you will only, the separation is based on molecular weight or mass of the molecule. The second important point, the same instrument, you can also analyze negatively charged molecules. So you can put into negatively charged time of flight mode, then you will allow only negatively charged molecules. Neutral molecules, non-ionizable molecules cannot be detected by this method. That is one of the limitations. So I have explained here now the principle involved in MALDI-TOF. You have a laser energy being supplied to the sample. Ionized molecules without getting fragmented will move into the time of flight tube and gets detected. Coming to the work, I had an opportunity to work in biological control agent, trichoderma. As most of you know, trichoderma is one of the most beneficial organism used in agriculture as a mycoparasite. It has got all the properties: antibiosis, antagonism, competitively high, fast growing, then direct parasitism. It can function and kill soil or seed-borne fungi. And hence it is being potentially considered, and many species and strains are exploited commercially also. Commercially, trichoderma is available. I had used it as a biocontrol agent. I did not know the array of compounds being produced. And when I went with the Indian Academy of Sciences fellowship to Professor P. Balaram's lab in Indian Institute of Science Molecular Biophysics Unit, I came across MALDI-TOF instrument there. Then I was looking for the literature. Only 4 papers in 2001 were available on bacteria, use of MALDI-TOF. Then no paper were available because most of the biologists, microbiologists will not look into the mass spectra journal. So it was getting extended to biology and particularly microbiology. Bacteriologists were ahead of mycologists and they had used it. And when I looked into the instrument, got into the literature, I found it is interesting. Then directly from this fungal culture plate, placed the spores and the mycelium into the MALDI. And the technician was not allowing me because the fungus was there. But I showed him they had done with the cells, nerve cells and blood samples having been done. So I asked him to do the fungal cells. To our interest, Professor Balaram was so excited to know the result. What was the result? So there are a group of compounds, antimicrobial compounds produced by trichoderma and related fungi. They are called peptaboles. Peptaboles. They are peptides, the small molecules having amino acid sequences containing AIB. AIB is a non-standard amino acid, non-proteinogenic amino acid, alpha amino isobutyric acid residue in more number. And you know, in any peptide or a protein, you have a C-terminal and N-terminal and C-terminal. C-terminal portion is an alcohol here. That's why peptide containing AIB and alcohol. So peptidobole. There are number of peptaboles available. They are antibiotic peptides produced by trichoderma, characterized by the presence of unusual amino acid AIB, and that C-terminus is alcohol. For example, if phenylalanine is there, it would have been converted into phenylalanol. Leucine is there, leucinol, isoleucinol, valinol, like that. Then they are highly micro-heterogeneous, differing in their molecular weight by 14 Dalton. One peptabole differs from the other peptabole in molecular weight by 14 Daltons. So why that micro-heterogeneity? Lot of chemical diversity is there in peptaboles. For example, instead of AIB, alanine can be there or valine can be there. Leucine-valine exchange glycine-alanine exchange, glutamic acid-glucine exchange will allow 14 to 16 Dalton variation in the molecular weight of these peptides. What is interesting? They are synthesized by non-ribosomal synthesis by peptidase synthetase enzyme. So that being the case, these sequences are not available in the protein database. So Birkbeck College, London, they have developed peptidase database. As of now, 314 peptidase databases are available and they are stored there. So what did I get? When I made the spectra, directly the cell being put into the as sample, we got this sequence, this spectrum. The x-axis shows MZ ratio, molecular weight charge ratio. The y-axis shows relative intensity. It is a qualitative method. Quantification is not possible, but relative intensity of different molecules can be seen. One of the strains of trichoderma showed 1700 molecular weight corresponding to 17 residue peptidase. That was very interesting. You see the peaks were rising like anything, 5,000 to 6,000 relative intensity. And these are, each peak here differs from one to another, 14 or 16 Dalton, indicating this is one peptidase group. So having said that, there was another trichoderma strain producing 17 residue peptidase and 19 residue peptidase. Two groups were found. Just directly cell analysis within 2 minutes, we were able to differentiate chemically one trichoderma from another trichoderma as far as peptidase production is concerned. Then the third one, this is a different strain which was producing 11 residue, 14 residue, that means 11,000 molecular weight, 14,000 molecular weight, 17,000 molecular weight. So 1700, sorry, 1100, 1400, 1700. They are very small molecules because they are peptides. This clearly demonstrated it was possible cellular detection of peptidases and differentiate chemical diversity for one strain from the other based on their ability to produce. Then after obtaining this spectra, immediately I checked the other strain was producing 14 and 17 residue. Immediately I checked the Birkbeck College peptidase database available. In Trichoderma hertzianum only, one species, extensive peptidase diversity have been detected. 59 peptidases have been identified in one morphological species, Trichoderma hertzianum. What are those compounds? You have a table here: trichorosin, hertzianin, trichorosin 2, trichorosin 3, trichorosin 4. Like that, they have identified 59 peptidases in Trichoderma hertzianum. What was my work then? Using MALDI-TOF MS, we got the molecular weight. And look at the sequence. Each molecular weight 11,060, 11,061, 11,074. That means there is a 14 Dalton difference or 16 Dalton difference. So these molecules were found in our strains detected using MALDI-TOF. This characterization of sequence and other earlier to my work was based on NMR purification, HPLC purification, and then sequencing, peptide sequencing. Here, directly cellular detection within 2 minutes was possible. So different groups of peptidases like 11 residue peptidases, 14 residue peptidases, 18 residue peptidases, 19 residue peptidases, all were detected. I used 35 isolates of trichoderma and found which one is producing which type of peptidase. So how the result identification was done? We already have a calculated molecular weight based on the sequence available. Then what was the observed molecular weight in MALDI-TOF? We compared and our isolates showed presence of trichorosin, hertzianin, trichorosin HAV, HA5, trichokindin. So these molecules were directly detected from the cell. What is the advantage? You need not have to go for purification process, long HPLC procedures and other things. Directly cell will tell you which type of antibiotic this strain is producing. So this we published in Fungal Diversity Research Series, edited by Professor Sridhar, and also the book Frontiers in Fungal Ecology. Then further, there was an interesting This was about the peptaboles from trichoderma. The other interesting was a protein called hydrophobins. Fungal hydrophobins are a very interesting molecules. They secreted as monomers, particularly aquatic fungi and some plant pathogenic fungi. And these monomers gets accumulated over the hyphal tip, hyphae, over the hyphae, making it get exposed to the water level. So floating is because of the hydrophobin. Hydrophobic surface is being out. Hydrophilic surface is attached to the fungal hyphae. So this particularly interesting proteins found on many fungi will help in attached to the surface of a leaf or at a float in the water in case of aquatic fungi. Trichoderma, we were doing the peptabole detection, but interestingly, we found one more peak at 7,000 molecular weight. When I say 7,000 molecular weight, I was looking for their protein. The 1700 or 1100 are peptides. Then we were looking at a protein. Then we found this hydrophobin protein detected by the same spectra at a molecular range, higher molecular range. So in order to confirm whether it is a protein or not, we grew trichoderma in Zepecdox medium, two different media, one containing N15 labeled nitrogen source. Ammonium nitrate you have generally in Zepecdox medium, that we replaced with a N15. A normal one is N14. And when you grew the fungus will add, whenever it synthesizes nitrogenous compounds like its DNA or RNA or peptaboles and proteins, it will add N15. Wherever nitrogen is there, one atomic weight gets increased, which results in molecular weight of the protein getting increased. For example, in the base of this spectrum, you can see 7383 is the normal hydrophobin protein detected in trichoderma grown in normal Zepecdox agar medium, whereas 7467 is the molecular weight, the same strain grown in N15 containing medium. That means using isotopes, we were able to know it's a protein molecule having 80 extra nitrogen. Each nitrogen will improve one atomic weight, and hence the molecular weight got increased. So we were able to detect peptides as well as proteins in trichoderma strains. So biochemical markers for differentiation of trichoderma strains was possible with the peptide antibiotics. Intraspecific variation among trichoderma, I did mention 35 isolates we had. We were able to look at the chemical diversity in terms of peptabole diversity, which suggests metabolic diversity. Then cellular level detection of hydrophobins are possible. One important point here, most of the molecular biology workers look at the differences at the DNA. DNA is the director for cellular functioning. But if you are looking at the functional aspect, DNA, a particular gene may be present, but not getting expressed. You will not know by molecular methods. Where the gene is present, it is getting expressed in the protein. So proteomics proposers are highlighting functional ability of the organism to produce a protein. So detecting a protein would be much more beneficial rather than going for a DNA sequence. I have finally a comparison of the different techniques available, but for time being, peptaboles we were able to detect. Intraspecific variation within trichoderma population was possible. Then trichoderma hydrophobin was also detected. We ventured this into one of my PhD student, Aspergillus terreus. He had all the Aspergillus, 6 different Aspergillus terreus, one producing an antimicrobial compound, the other 4 are not producing a antimicrobial compound. So this trichoderma, we separated based on mycelial protein profile. And as you can see there, one of the isolate, this band is missing. That means that protein is absent. So for that, you need to extract the protein, isolate the protein and run on a gel and distinguish the different isolates based on protein profile. So what did I do? Again, you went for MALDI-TOF. One can see the cellular extract. The cells as such were not giving. So buffer extracts and after dialysis, the salt component was removed and it was used as a sample here. Then you can see here 12,494, the peak was absent, which is present in one strain, absent in another strain. That means what? A simple buffer extract of the cell was able to distinguish a protein molecule, because 12,490 is the molecule which is available in the first 3 peaks there were absent in the other strain. So within Aspergillus terreus, we were able to distinguish based on the molecular spectra obtained by MALDI-TOF. Likewise, all the 6 strains we were able to do. Then an extension or a modification of this work is another electrospray ionization mass spectra. Though overall principle is same, the method of ionization is by spraying minute drops and making it ionized. So one of my student recently got his PhD, Dr. Narendra Babu. We were working on pigments from different Aspergillus, Penicillium and Talaromyces strains. You can see some secretory pigments, some cellular pigments. So we had isolated these and then detected by regular UV visible spectrometry the compounds present, the pigments present. But later when we analyzed by electrospray ionization mass spectra, and the spectra obtained was subjected to progenesis software, we were able to detect some of the pigment molecules already known. This is the spectra. We could identify a molecular weight 227, which corresponded to ginipin, a blue colored pigment produced by Aspergillus nudulans. Then diazen, an yellow colored pigment produced by Aspergillus nudulans. Likewise, tetrahydrocurcumin, an yellow colored pigment with a molecular weight of 373, was detected in Talaromyces ruber. Interestingly, 3 different strains of Talaromyces ruber, we were able to find different molecules using mass spectra. So the other one, juglone, a reddish brown pigment produced by Talaromyces funiculosus. So far I have discussed the Aspergillus terreus, the pigments produced by fungi, peptoboles from trichoderm. Now I will extend the topic for other applications in microbiology, but specifically mycology. Say if you have a mixed sample like a blood culture, lot of things are there. So there may be many infections, or urethra or vaginal exudate, there may be many infections. So what is important here? To get the sample, directly load it. Then you will get different spectra obtained from different organisms associated with the sample. Then you need to have a software to check. The software will check which organisms are already there, characterized by MALDI mass spectra and available as a software. So your spectra is matched with the software and you will be able to detect. One such automated MALDI-TOF for identification of microorganisms is Biotyper. Biotyper is a software associated with the company called Bruker Daltonics from Germany, which supplies the instruments. So what they have done, all possible 4,000, 5,000 bacteria, they have isolated, obtained the mass spectra, and they have kept it as a model. So whenever you get an organism, do MALDI-TOF and compare with the already existing spectra to know which organism it is. Then another company called Diagnostic, they came out with a spectral archiving and microbial identification system, SARA-MIS, another dedicated software for microbial identification. You get the sample and compare. How to do that? You have a unknown colony from a plate, culture plate. You can pick up the colony and plate it on MALDI-TOF and get the spectra. The spectra is compared with the already identified organism spectra, and software will tell you which organism is this. The same thing is extended now. Earlier they were interested only with the bacteria. Now it is extended to molds, multicellular fungi and yeasts also. So the Bruker introduced novel fungal identification system, MALDI Biotyper, and because colony morphology, there are other problems with the microscopic cultural conditions and things like that. stable character is required. So they developed what is called mycelial transfer technique as a sample preparation. What is interesting here, directly only 2 microliter of the sample is enough. So you need a fungus growing, which is moldy biotyper system. The filamentous fungi, this was the mycelial transfer technique, what they developed. They used a toothpick, wooden toothpick, to pick up formic acid droplet and touch the fungal mycelium. So formic acid will react with the fungal mycelium, releases the proteins, and that is plated on MALDI-TOF plate, sample plate. And then using a laser energy, matrix is added there. Using a laser energy, you ionize the molecules, which can get detected at the final stage. So what are the current applications? They are rapid. The technique is rapid. Sensitive, only 2 microliter of the sample is required. Simple method for characterization of the molecules. Sample preparation is less than 2 minutes per sample. Analysis time is about 5 minutes. Identification of strain, species, and genus-specific biomarkers by direct cell analysis, single cell analysis. The laser beam hits at 2 micrometer. Most of the fungal spores are bigger than 2 micrometer. So you will have only a single cell also can be analyzed. Continuous monitoring of the air can be done in a food or pharmaceutical industry. Microbiological analysis of food and water can be done. Medical diagnostics, facilitating rapid screening of hundreds of samples which you receive in a diagnostic lab. Chemotaxonomic classification based on biomolecular profiling. You may not identify directly what is that compound is, but you will be able to differentiate one strain from the other based on the presence or absence of a particular molecule. Detection of proteins, peptides, nucleic acids, phospholipids. But phospholipids and DNA are poorly ionizable in these conditions. So mostly used for proteins and peptides. Analysis of fungal metabolites, interestingly, mycotoxins, aflatoxin have been detected directly, and it was possible to differentiate toxigenic, non-toxigenic strains. Analysis of cyanobacterial metabolites like microcystins, direct peptide profiling. Not only detect the peptide, you can also identify the amino acid sequence of the peptide. So peptide profiling can be done. So did I mention all these applications directly without any proof? I am going to go produce the proof for you. Rapid identification of filamentous fungi. Look at this: Aspergillus flavus, Aspergillus fumigatus, Trichophyton rubrum, Rhizopus macrosporus, Microsporium canis. Medically important fungi have been identified to the tune of authenticity up to 92.7%. They had 123 filamentous fungi. Peng et al. 2019, the reference. 123 fungi. Out of that, 114 fungi have been identified properly. Few were Mycelia sterilia, so they did not have proper identification done by microscopic methods, but the accuracy was 92.7%. Then how to do for a filamentous fungi or yeast? So you have fungal identification problem, one or few species you have, go for MALDI mass spectral biotyping and compare with the other barcoding or metagenome or other Sanger sequencing, qPCR, loop mediated isothermal amplification. They have compared. Drusiner and Fremuser 2017 provided this for fungal analysis. MALDI biotyping, species identified. Species not identified by MALDI, then create a new spectrum, reference spectrum. You have a new organism, new strain, which reference spectrum is not available. You can create and correct the species, upgrade the software. So mass spectrometry based proteomics analysis for detection of plant pathogens people have done. Then extracellular expression of recombinant Ganoderma protein. Recombinant fungal protein expressed in Bacillus has been identified using MALDI-TOF. Then Penicillium expansum, Penicillium digitatum in apple were distinguished. That means agriculturally important fungal analysis. Then in medical mycology, lot of methods have been developed recently. This is one of the recent reports, Robert et al. 2021. published in Microorganisms Journal, which provided a common yeast identification or common filamentous fungal identification, rare fungi identification, and prepare the software and database dependent. If a new organism is there, that has to be identified by other methods, and then software can be upgraded. Fungemia, the presence of fungi in blood cultures, also being detected here by processing the blood sample directly for MALDI-TOF. Then typing approaches, antifungal susceptibility tests, all being done using MALDI-TOF. So it is a mechanized system. ATCC and MTCC cultures, now facilities in American type culture or in tech, they have got MALDI-TOF, and this technique is yet to come in detail with the books. Then medical mycology laboratory, one of the recent reports, the implementation of MALDI-TOF revolutionized practices and significantly reduced the identification process of yeast and filamentous fungi. With the onset of COVID, you have mucormycosis and other, generally being referred as black fungi, but it was possible with the MALDI-TOF, the identification process being reduced, time being reduced, and fast analysis. Significant advances have been made in MALDI-TOF analysis for species level identification of yeast. I'm sure most of you know the yeast very difficult at microscopy level, but MALDI-TOF within 5 minutes it was possible species level identification and achieved the clinical laboratory results. So purification of an antifungal protein from a plant, antifungal protein from a plant has been identified using MALDI-TOF. Then in veterinary science, veterinary practice, a panel of 290 mold and yeast isolates representing 69 different fungal species was isolated and subjected to MALDI-TOF and conventional methods. The performance of the two methods were compared and confirmed by DNA sequencing. Then it allowed 89% of correct identification. For DNA sequencing, you need to isolate the DNA, then go for sequencing. Here, you just need to take the organism or its extract and identify. The identification accuracy is up to 89% achieved. Baker et al. 2019. Then 32 fungal isolates from 80 food samples with the accuracy of 94.4% was achieved. Alternaria alternata, Aspergillus niger, Aspergillus flavus, Penicillium digitatum, Candida albicans, Deboromyces, all these were identified as a foodborne fungal pathogens using MALDI-TOF. One interesting paper I found yesterday when I was looking, published in 2022, the MALDI-TOF mass spectra can differentiate Aspergillus flavus toxigenic and non-toxigenic strain. Very interesting, toxigenic and non-toxigenic strain. They created a condition to get a peak, which I have showed in arrow, 335 molecular weight corresponding to aflatoxin. They did aflatoxin separately, they did the fungal cells separately, culture filtrate separately, TLC purified aflatoxin, all that. Then they found a producing strain, the lower half of the spectra, showing 335 molecular weight corresponding to aflatoxin. So directly by cell analysis, if you can detect whether a particular toxigenic strain is there or non-toxigenic strain, what you get. So none of the pepper or groundnut varieties in India are resistant to Aspergillus flavus. So most of the pepper samples comes back from, when it is exported, comes back from US and other countries saying that aflatoxin is there. Here is a rapid method to know the fungus which you found on the pepper or groundnut, whether it is aflatoxin producing or non-producing. MALDI-TOF will tell you within 5 minutes. So it was an interesting paper to detect not only fungi, but also fungal metabolites like toxins. Then MALDI-TOF methods based in foodborne yeast isolates. Then dermatomyces, an enzyme metalloprotease produced in dermatomyces was characterized using MALDI-TOF. Then environmental samples. You want to isolate let isolation part being left out. Directly isolate the from air or water, and what we do like metagenomics, you can go for MALDI-TOF. So 149 isolates 18 yeast, 131 fungi were analyzed, and they were able to get 92% accuracy in MALDI-TOF isolates correctly identifying. This is one of the recent paper where fungal allergens are being checked. So antimicrobial compounds, peptides produced by a frog has been identified against pathogenic chytrid fungus. So a lot of work has been extended from the past 2 decades. So microbial species biodiversity in the settling dust within. So you have a air sampler, it sucks the air and then place it directly on the MALDI-TOF plate, and then where you can process it. So they have compared MALDI-TOF biotyping with LAMP assay, Sanger sequencing, NGS, and this was found to be better. But experimental cost as machine, this is higher compared to qRT-PCR. So that is one of the limitation. For example, the MALDI-TOF instrument produced by Bruker costs 2 crore rupees. So that's why every laboratory cannot have. Indian Institute of Science has got a national facility where one can get the sample processed. So now IMTech, CCMB, BARC and ATCC, they all have adopted for MALDI biotyping process of identifying the organisms or the metabolites of interest. Then I have shown you here an expanded version of applications of MALDI-TOF for mycology. But what are the limitations? One, as I mentioned, the instrument cost. Instrument costs 2 crore rupees. Not necessary that every institute, every university should have. One can go to an institute where we can get our sample processed. So, in nutshell, it has opened up extensive functional ability of fungi to be identified, having applications in medical field, veterinary field, food industry, biotechnology industry, antibiotic screening, and all that, as evidenced by these publications. So, what is in conclusion? Characterization of biomolecules is possible. The microbial strain-specific molecular profiling help in identification and diagnosis. Biomolecular profiling for biodiversity assessment, even at intraspecific level. Then this technique can be used for the detection of microorganisms, plants, and animals, extending the horizon of application. The limitations are many. You cannot identify non-ionizable molecules. You cannot identify directly what molecule it is, but you can identify microorganisms if the software is having the reference spectra. Otherwise, go with the conventional and biochemical methods of identifying an organism, a yeast strain or a filamentous fungus strain, and then upload or make the spectra available in the software. Then you will be able to compare the new organism to identify. This will help you in functional detection, because a protein or a peptide or any other is the expression of a particular gene, and this, the time required, is an additional advantage over other molecular methods of identification. So it has opened up a new rays of light for mycologists, microbiologists, and biologists. A physical method has been adopted in biology, and day by day it is expanding. Mine was the 4th paper in mycology, but now you have hundreds of papers in mycology. But bacteriologists are ahead of as far as using MALDI-TOF for the analysis. So it is essential for me to thank the people who are involved. Professor B. Balaram, my teacher fellowship guide, Professor Deepankar Chatterjee, in charge of director of the molecular biophysics unit, proteomics facility. The Subramania Prakash was my friend and a technician there. And I thank Indian Academy of Sciences for providing the teacher fellowship, and my research scholars K. Sandesh and Dr. B. N. Narendra Babu for having performed the experiments which I used here. And thank you, MycoAsia, for providing this opportunity. Thank you very much. I look forward for questions.
Thank you, Dr. Shishupala. Very nice, informative talk. So moving on to our question answer sessions, I think Dr. Shenoy has a couple of questions. Dr. Shenoy?
Yeah, thank you, Rohit. Am I audible, Rohit?
Yeah.
Professor Shishupala, can you
Yes,
hear
sir.
me?
Thank you, sir.
It was a wonderful presentation with loaded with basics, applications, limitations. I wanted to probably share a couple of insights from my personal experiences. I mean, you stated that this tool can be very rapid. It is very simple to use. But my point is it cost effective? Is how accurate is this? Because
Yeah.
it is a great tool probably to characterize a set of molecules present in the fungus, but can it actually taxonomically identify that fungus? Because taxonomic identification is basically comparison of your fungus with the type material or
Yes.
the sequence molecules from the type culture, for example.
Yes.
I'm sure you might have already thought about it. Maybe you could share more light on it, please.
Yes, sir. Yes, sir. Sir, if it is already a typed fungus, identified fungus by other means, it can be used as a reference spectra for unknown organism to be compared. So as you rightly mentioned, in case of fungal identification, the colony morphology, microscopy, and others will vary depending on the type of media, culture conditions which you have used. But here, you need not have to identify the molecules, what molecules they are. It will, all the ionizable molecules in the typed fungus, identified fungus, will be displayed as a spectra. Then it is possible, if it is used as a reference spectra, a new organism, new isolate, missing a particular peak, missing a particular molecular mass, or showing an extra molecular mass, one can go for either species identification or for, so you need to have a bioinformatics tool like software attached to this instrument for microbial identification process. So species identification. The first part of the question was cost effective. See, the chemicals which we are using, the instrument only machine cost is high, up to 2 crore rupees. But the sample processing per day, you can process 400 samples. When you divide the running cost, like it will be coming less than 5 rupees or so per sample. So only you need 2 microliter of the sample, then 2 microliter of alpha cyano, the matrix. So the running cost would be less, but the initial investment for the instrument is more. That's why I did mention that limitation can be overcome by using national facilities available in our country.
Thank you very much. I will let the audience ask question. Dr. Rohit, please.
Yeah. Thank you, Doctor. I think Mr. Deepak, you had a question. You had raised the hand. Mr. Deepak, are you still there? Mr. Pawan Reddy?
Sir, by using the MALDI-TOF, can we identify the mycoviruses?
Mycoviruses, yes, they have done virus identification. Mycoviruses, it must be possible because when they have done for corona or for HIV and other, the bacteriophage identification they have done, it could be possible. I need to refer for mycoviruses.
Okay, sir. Thank you.
Yeah. So, Dr. P.N. Singh, you had raised your hand a little while ago. So, do you have your question?
Sir, myself Deepak Kumar.
Yeah, Deepak. Yeah, go
Here
ahead.
I am asking one question, one or two queries. If fungal identification is similarity with shows the similarity 94%, then how we confirm the particular fungi? That means the species of that particular genus.
I think you took it in a different way. 94% from the reference saying that they had 125 fungi. Out of that, 114 was identified correctly. So the other Mycelia sterilia, because they did not know what was the isolate in the regular process, they were unable to identify. I'm not telling 94% in that reference I did mention. 94% correctly identified. Rest was not. So for individually, you need to make the reference spectra possible by making, there will be individual errors when you put the sample concentration, matrix concentration, then the amount of laser hit ionizable molecules. So whenever a software is developed, you need to have a reference spectra having, let us say, 10 isolates of Aspergillus flavus. So
Yes.
consistently, if you are getting a particular mass peak, for example, as I showed, in case of trichoderma, we were consistently getting peptobole sequences. So such characteristics peaks, if you have, then identification is 100%. You cannot just directly say MALDI-TOF, provided your reference spectra is ideal, you can relate it to either species level or genus level identification.
Actually, sir, I am working in NFCCI, National Fungal Culture Collection of India, and recently we got a MALDI-TOF analyzer.
Yes.
And here I am unable some fungal cultures identification on the basis of MALDI. So my simple question is that how many year old of that fungal cultures are required for the MALDI-TOF?
How many hours old or year old?
How many days old?
Time. See, I did mention trichoderma within a day, the spore directly being put, or in case of some of the yeast, directly the skin sample from dermatomycosis sample is directly put on the MALDI. So there is no need for complete growth of the organism and complete petriplate or few days. So in certain cases, directly it must be possible to do directly clinical sample also without growing the fungus. Like you have a metagenomic approach, you have directly the sample being processed and compare with the reference spectra.
Okay.
If I can just add to it, to the question of Mr. Deepak, Dr. Shishupala, I think, sir, there is some kind of score also there, 2 score, 3 score kind of thing, and we have to match the peaks.
Yes.
Yeah.
Yes. In MALDI-TOF, you have a camera also to see the sample which you have placed. So the spore, I did mention, the laser hits only 2 micrometer. So a single spore or a single hyphal thread is sufficient enough, provided if the molecules are ionizable under the conditions which you give. So many molecules may not get, it is not giving complete molecular profile. So ionizable condition, if it works well with the organism you have, then you have the identification done.
Sir, there is any extraction protocol for the extraction of that particular compound?
Yeah, I did mention alpha cyano is the matrix preferred for
CCA matrix.
Yeah, CHCA matrix is preferred for peptides and proteins. So
then, sir, there is a range limit, 2000 or 20,000, some range limit between this.
Yeah. That also you can fix. Now MS up to 12, M to the MS to the power of 12. You can go on restricting the molecules. You can look for small molecules. You can look for peptides. You can look for bigger proteins also.
Okay, thank you, sir.
Yeah, Mr. Deepak, you can also look for the, I think, Dr. Shishupala, is there formic acid also sometimes used for?
Yes, sir. Formic acid, synapinic acid, gensic acid, these are all being used. See, I did try earlier with Fusarium and Rexclera. They were not giving readily peaks. So in such cases, you may have to use an ultrasonicator to break open the cell and get the cell extract. So it depends on the fungus, how rigid it is to give ionizable molecules under the conditions provided.
So,
sorry to interrupt, Rohit. Dr. Shishupala, can you please switch on your video? Rohit, please go ahead.
Yeah. So next is by Mr. Jose. Mr. Jose. Mr. Jose. If Mr. Vinay Halor.
Good morning, sir. This is Vinay speaking. Am I audible, sir?
Yes, you are.
Sir, I am a medical mycologist working at AIMS. Very nice presentation, sir. Really
Thank you.
appreciate the effort taken by you. And sir, I just have to add to your presentation that the papers that you mentioned, wherein they have shown that they were able to detect the fungus directly from the sample, they are in research mode only, sir. They
Yes.
they need to be more rigorous to be applied in clinical setup. Reason being, MALDI shows some problem when there are more than two organisms. There's a mixed mode also,
Yeah,
where
yeah.
we can find out the limit of detection of MALDI is two organisms. So the problems we as clinical microbiologists face is that many of our samples have more than one organism. So MALDI can differ at this point in time. The Bruker MALDI, the one that you described in a presentation, can differentiate between two organisms. There's one more system called as the Vitek MALDI. I'm not sure if the Vitek MALDI can even differentiate more than one. So these are the problems that we as clinical microbiologists face. And another issue that I faced long back when I was working at PGI Chandigarh with MALDI was we used to have this problem with dematiaceous fungi.
Yes.
They cannot be identified with the regular on-plate extraction. We have to be very careful, catch the hyphae when they are young, because the moment they start producing pigments, the biotyper will not be able to identify it. And as Dr. Rohit mentioned, uh, the MALDI biotyper has a cutoff. So if the cutoff is 1.6, you can identify it till genus level. If it's more than 1.8, then you can identify it up to the species level. Now
Yes.
the problem also comes when you have species complexes. So when there is a species complex, and the problem also comes especially in dermatophytes, wherein the original type material only is missing, and the nucleotide databases are also corrupt. So Vitek and Bruker MALDI, both of them we have personally found that they will confuse Trichophyton interdigital with the Trichophyton mentagrophytes.
Yeah.
And those problems are there. They are practical problems, but a very wonderful presentation, sir. It was very, I mean, I would say that my daughter, who is 10 years old, would understand it better than me. It was beautifully made, and thank
Yeah,
you for
yeah.
the very simple presentation, sir.
Thank
And
you. Thank
probably
you.
it comes from first-hand experience,
Yeah.
because you were one of the first few persons who worked on MALDI. Beautiful presentation once again, sir. Thanks a lot for taking your time out and explaining it so nicely.
Thank you. Just
Thank
to
you.
supplement,
Yeah, yeah.
I did mention dematiaceous fungi like Carularia, Drexlera, those complexes very difficult to ionize. So now you rightly said biotyper, Saramis, they are also evolving. Earlier they were concentrating mostly on bacteria. It was working wonderfully well. When they extended it to fungi, you have a spore, different molecules being present. You have a mycelium, different molecules being present. In clinical samples, you have related, unrelated fungi being there. So Biotyper and Saramis and Vitek, they are also improving day by day in making better reference spectra. In days ahead, it must be possible. I wonder how did this is yet to come in microbiology books. You have
Yes,
only
yes, yes,
latest books also having
yes,
a paragraph which
yes,
I have worked 2 decades back.
yeah,
So
yeah,
it
yeah.
was really looking at the applications. It must be possible. You can extract formic acid, ultrasonication, try out different methods of making the molecules ionized. Thank you for your compliments.
Yeah, thank you, sir, and thank you, Mr. Vinay, for the. And I just have a small comment. I think I echo Mr. Vinay's comments that it was so nicely animatedly explained that it was very easily understood by others. For the one of the comments that I wanted to make was regarding the extraction of the mycelia and that thing. I think as the time will progress, the database will be added and then it will be going. So moving to next, Dr. P. Shivakumar Singh.
Sorry, please wait. Mr. Jose.
Namaskar, sir. I am Dr. Shishupala from Telangana State, Palamuru University, Department of Botany. Here we have applied mycology specialization for botany.
He was muted.
Sir, greetings, sir.
Yeah, Dr.
Myself,
Shivkumar
Dr.
Singh,
P.
please repeat.
Shivkumar Singh, Dr. P. Shivkumar Singh, sir. I am from Telangana State, Palamuru University, and in MSc Botany, molecular mycology specialization here, sir. So our students directly learn this.
Can you be
class.
little louder,
I
Mr.
have
Singh?
sir.
Yeah.
Hello, sir. Are you getting
Yeah.
my voice, sir?
Yes,
Yeah, you be louder, please, or put it in chat box.
Yes, sir. Okay.
Be specific.
And I thankful to the MycoAsia and MycoIndia, sir. Our student directly this class has telecasted in our class, theory class. Thank you.
Yes, thank you.
And another one doubt I have, I will post in box. Thank you.
Thank you, Dr. Singh. Thank you. That is much appreciable. I think Mr. Jose had raised her hand earlier, and we missed him earlier. Please go ahead, Mr. Jose.
Sir, shall I take up some questions from the chat box?
Sure. So there are only one remaining for the raised hands, like Surbhi Sharma, and then we'll go with the chat box, sir.
Okay,
Sir,
fine.
Yeah. Surbhi, please go ahead with your question, then we go with the chat box.
Okay, sir. Thank you, sir. Thank you, sir. It is a wonderful topic, informative session for as a beginner. I'm a beginner in mycology department, and I work on identification and antifungal susceptibility pattern of fungus isolated from nosocomial infections. And my question is, MALDI-TOF procedure for extraction of any diatomaceous fungus or any yeast mold. Is there any kind of SOP are available? According to SOP, we will perform the MALDI-TOF.
If it is a known organism, it is available. Already published information is available. What is the procedure for processing for MALDI? If it is for an unknown organism, then you need to formulate like one sample directly, one sample cellular extract, another sample ultrasonicated, another sample formic acid digested proteins and peptides, like that. Some 10 types of samples you prepare, and then hit upon one which is giving you consistently good result, or the spectra.
Okay, sir. Okay, sir. If there is any SOP are available in soft copy, or if there is any link available, please kindly share in a group, so I will further then read.
I had put most of the references. If you have not noted, you will be shared with the literature.
Okay, sir. Okay. Thank you, sir. So much informative session. Thank you so much, sir. I'm looking forward so many sessions, MycoAsia and MycoIndia. Thank you, Rohit, sir. Thank you, all the MycoAsia members. Have a great day. Thank you.
Hello.
Hello, sir.
Yeah, Dr. Rohit.
Yes, sir. If you, Dr. Singh, you are audible.
Yeah, sir. Good morning, sir. Shishupala, sir. Good morning.
Good morning.
Actually, it was a very nice, wonderful lecture. Even I can learn something from this your lectures. And I have a very silly questions because I am a hardcore mycologist here in this Agarkar Research Institute and FCCI. So in nature, many fungi, this like a microfungi, while collections, I'm seeing some very colorful exudates on that surface of this mushroom fruit body, and even in sometimes rarely in cultures also. So can this molecular this MALDI through MALDI, if we take that exudate, so is it possible to analyze few things for that some chemistry?
Yeah. This is an interesting part. When we processed actually some of the pigment, fungal pigments, melanin is not easily ionizable.
Okay.
It will prevent other molecules also to get ionized. So a modified version of mass spectra, electrospray ionization, ESI MS, through which we got fungal pigments, particularly from Talaromyces, Aspergillus I explained. ESI MS is possible. So there are different variations of mass spectra. If it is one is not working, then you may have to try with the other. Common question being asked is whether identity of the compound or metabolite is possible. No, directly it is not possible.
Okay.
Because this technique will give you only molecular weight of the compound associated with the sample.
Okay.
So you need to know or already identified molecules, its molecular weight, and whether it is there in my strain or the species, we need to check. One question was there whether molecular metabolite identification, I did show in pigments, we got the electrospray ionization done, but the peaks obtained were subjected to a progenesis software, which gives identity of the metabolite if it is already known. If it is a new one, you need to go for all biochemical characterization.
Thank you, sir. That was my Okay, sir. Nice.
Thank you.
Rohit, unmute yourself, please.
Sorry. For Sheetal Chaudhary has asked that for a species with unknown metabolite diversity, how its identity will be detected.
I did mention biochemical characterization is required for metabolite identification. One of the biophysical parameter, the molecular weight of the compound, you will get here. But if it is already known in a reference spectra, then you can identify whether this is producing. For example, I did mention 335 is the molecular weight of aflatoxin. So whether toxin is there or not, you can detect. But if it is a new metabolite, one has to go for NMR, biochemical characterization, and then use it as a reference.
So thank you, sir. For next is Kalpana Survanshi has asked that if fungal identification by MALDI-TOF is more difficult as compared to bacteria, and if so, what can be the troubleshooting steps to improve the clinical samples?
Yes, definitely. See, in case of bacteria, they are single-celled, uniform. The chemical composition of the single cells are uniform. One of the troubleshooting in case of fungi, you have vegetative hyphae or mycelium with the spore structures conidiophore or spores being different in their molecular composition. So you have though the conidiophore is a specialized branch of hypha producing the conidia, but the spore chemicals are different from that of mycelial chemicals composition. The secreted molecules are different. So whether you are taking from the medium or you are directly doing the cell analysis, you need to be sure whether you are taking vegetative hypha or a spore or the secreted compounds. In some of the fungi, our experience shows all the molecules not necessarily be present in the cell. Some are secreted into the medium. Many of the people, though I do have an objection, for example, many of the people use PD broth, potato dextrose broth. They simply forget that there is a potato extract. The compounds be present in the potato extract after cooking will be interfering with your analysis. So I would prefer a Zepecox, a synthetic medium, where there is no natural ingredient available to grow the fungus in order to analyze. So one of the troubleshooting is morphologically distinct. The mycelium or the conidiophore, the phyllides or the conidial head, all may have chemically different because they are morphologically different. Obviously, it comes from the differences in the chemical present. Though mitosis, we do refer for conidiogenesis, there will be different molecules present in the hypha, different molecules present in the conidiophore stalk, and different molecules present in the conidia, unlike bacteria. Whereas in case of bacteria, of course, endospores, spore chemicals they have identified. The spore chemicals are different from vegetative cells of bacteria. Except the spore formers, most of the bacteria have got ready single cells uniformly maintained. But in case of fungi, you need to work out the different sample which you are taking. So formic acid extracts for proteins or a matrix you need to come by combination together, whether you are taking only cells like spores or zygospore or a sexual spore, like that you need to work out with different samples possible. In some cases, cell lysates will do the work for you.
Rohit, unmute yourself, please.
Sorry. So another question was by, I think it was,
Biochemical markers which can be
Yeah.
used to differentiate between strains.
yeah.
We did show peptobole markers in case of trichoderma. As I showed the spectra, 11 residue, 14 residue, 17 residue peptoboles were differentiated. So it is important with which, for example, as one of the reference which I showed, aflatoxin producing strain from non-producing strain, which could be differentiated. Like that, it must be possible. You need to work out which strain you are working for, which metabolite you are looking for.
Thank you, Dr. Shubha. I think there is one raised hand by Dr. Singh. So, Dr. Singh, can you go ahead with your question? You have been waiting for a long time.
Thank you, Dr. Rohit. I am Suresh Kumar from Manipur University, Department of Botany. Can you hear me?
Yes, we are audible.
Yeah, I just wanted to know, Sir Shishupala, where are the institutes which are doing this analysis, means on payment basis, so that we can send some samples, and how is the procedure to prepare a sample for sending? Can we send a live sample or the extracted sample?
In Indian Institute of Science Molecular Biophysics Unit, in their website, they have a proteomics facility page, separate page, proteomics facility. There is a sample submission forum, and that has to be downloaded, filled with the details required, and the sample need to be posted there.
Okay.
Right. And depending on where you are working, what for you are working, they may charge you, though it is a national facility. If you are from an industry, they may charge you more. If you are from academia, they will charge you less. Whereas in my case, I was working there only. So I used to take my sample from university, I go to Bangalore Indian Institute of Science and directly do it. Even we have done with some endophytes also. So there is a forum available as far as IISc is concerned. In other institutes, you may have to explore how do they provide the facility.
Okay, thank you, sir.
If I can add to it, Dr. Shishupala sir, that Dr. Singh, what I know about is NCMR was providing a service a couple of months till ago. At present, they were in the, I think, shifting mode. So they were providing at the charge of around 250 to 300 rupees, and you can directly send the samples to them for the analysis kind of thing, as Dr. Shishupala has mentioned that. So it's very economical kind of thing. I just have one point to raise, sir, that a few minutes ago we were discussing about the directly blood samples. I think if it is able to successful, then it will be very nice for the neonatal sepsis kind of thing, because neonatal sepsis, the time period is very less to detect the organism and start the treatment. So it will be a very benefit for the kind of.
Yes.
I think we shall skip the other questions, because there are a lot of questions, and if you can just put those questions on the MycoIndia or MycoAsia WhatsApp group, it would be nice of that thing. So I think thank you sir, thank you very much, Dr. Shishupala sir, for the nice talk. I think you had clearly informed us about the complete process of MALDI-TOF. It very nicely, with the basics of, even in a PowerPoint, you were able to explain very nicely. Even I didn't know very nice multi-process. And also you had mentioned about given the applications and the usage with the examples, especially with the examples. So it was very nice to learn about those things. And comparing the peaks, how one can compare the peaks and identify different kind of thing. And you had very amazing results for the trichoderma, because the morphology one cannot identify it, but you have a very different kind of peaks and other data that is coming from those things. I really agree with you that Professor Balram would have been very excited to see the first results of the fungi at that time. So definitely the technology has come a long way. It is in the process of improvement, and all those hurdles that earlier one of the person who has asked the questions was mentioning, those hurdles will be removed in due course of time. And definitely new database is getting added. Bruker is also adding the database, and you can also add new database into it with they're having. So I think and probably I think Dr. Shishupala, it's a very economic service as far as service is concerned. It is
Yes, sir.
even
Yes, sir.
cheaper than the sequencing. Sequencing will be 1,500 or 2,000 rupees.
Yes.
And as a service, it is maybe around 200 to 500 rupees per sample. Yeah. So,
sir, just I wanted to share some 2 minutes.
Yeah.
Most of the youngsters are there. The slide which you were mentioning, the two slides where I demonstrated positive ions, negative ion, positive ion moving in the time of flight.
Yeah.
It was developed in 2002 by me and my wife.
Okay.
For the youngsters here who simply download and copy, this two-slide preparation took me 18 hours. 18 hours.
Yes.